Good afternoon, everyone. Just thank you for being here. Also, just a good afternoon to whoever may be streaming live. My name is Pakou Vang. I'm a third-year pharmacy student at the Gregory School of Pharmacy. And today I'll be presenting my research entitled Assessing the Influence of the SLC2A9 Polymorphism on Sugar-Induced Hyperuricemia. So these are just some abbreviations that you may see throughout the lecture. To start off, I'd like to just provide a brief background. The major source of added sugars in the American diet is sugar-sweetened beverages. Since the 1970s, introducing additional sweetener other than table sugar, high fructose corn syrup is a major source of added sugars as well, and that's found in commonly consumed soft drinks or commonly consumed beverages, including soft drinks, energy drinks, fruit juice, and even coffee and sweetened tea. Daily consumption of sugar-sweetened beverages can increase cardiovascular or mortality risk by cardiovascular disease by about a third. And frequent consumption of added sugars has also been shown to be associated with the development of many common diseases such as diabetes, gout, even heart disease. Sucrose is a disaccharide that is composed of two sugars and is degraded in the body into fructose and glucose, which then get absorbed into the body. Fructose is metabolized in part to uric acid, and despite the structural similarity between the two sugars, fructose actually undergoes a different metabolic process as compared to sucrose. And then I also just wanted to note that uric acid has been noted as a biomarker for cardiovascular disease. So this is just a diagram showing the metabolic processes for fructose. Glucose is not illustrated in this diagram here, but if you look at the top, this is fructose here. Just a quick summary of the metabolic process for glucose. It pretty much is similar to this here, and it undergoes breakdown via hexokinase, which is the enzyme, and then it gets converted to F6P, which is fructose 6-phosphate, which then proceeds down the rest of the metabolic pathway. Now, with fructose, as you can see here, this pathway that I just went over for glucose, this is a minor pathway for fructose. So instead of that pathway, fructose gets metabolized via this enzyme here, and it follows this pathway here into fructose 1-phosphate, and then it gets degraded down further and ultimately gets converted into pyruvate, which then goes down the glycosidic pathway. And I wanted to note that the significance of this difference in metabolism is that because it doesn't undergo this pathway here, in its metabolism, it produces uric acid as a byproduct. So that's just mainly the highlight here. And yeah, I have fructose highlighted there in the red box. And then it produces uric acid as seen there. So acute hyperuricemia has been attributed to polymorphisms in a specific gene, specifically the SLC2A9 gene. This is a gene that encodes for a high-capacity urate transporter, specifically GLUT9 or glucose transporter 9, and that's found primarily in the kidney and in the liver. This transporter is known to be 50 times more effective as a urate transporter, although it can also help to facilitate transport of glucose. It plays a major role in maintaining homeostasis of uric acid levels within the body, and uric acid acts as an antioxidant within the blood, protecting just the body from damaging effects of free radicals. However, we want to maintain a balance of urate levels within the body, because if you have too much, then that can cause development of diseases such as gout. But if you have too little, then you're losing the protective effects of the antioxidant properties. So just wanted to throw that out there as well. And carriers of the variant in a specific polymorphism RS11942223, as listed there, has been shown to or individuals with that variant have been shown to have elevated blood urate levels following a fructose load. So that is the basis of our study, which I will discuss in a little while here. And lastly, I just wanted to note that saliva has been shown to have similar urate levels as compared to serum or plasma of the blood. Therefore, we decided to go with this route of collection for our study. So the primary objective of the study was to determine whether the SLC2A9 gene, or polymorphism, impacts systemic exposure to urate. And the primary outcome of the study was a change in salivary urate levels up to 4 hours following the fructose load. So now I'll just go into a discussion of the methods. Our participants were recruited either in person or by email. A demographics and general health questionnaire was administered. That was just to obtain baseline information and background information, and as well general health information on the participants. Following an overnight fast, saliva was obtained for baseline uric acid levels. Then participants consumed a 300 milliliter solution containing fructose and glucose as indicated on the slide there. And then additional saliva samples, approximately a quarter of a milliliter, was collected at each of the following intervals after the fructose ingestion. So at 30 minutes, 1 hour, 2 hours, 3 hours, and 4 hours following the load. And then uric acid levels were measured by enzymatic assay. Saliva samples were also used for DNA extraction, and genotyping was done using the TACMan RT-PCR assay, and that was to determine whether or not the participants showed the presence of the variant. And then just also informed consent was received for all participants in the study. A statistical analysis was performed using a student's t-test, and a preset alpha level of 0.05 was determined to be statistically significant. This is just a screenshot of the general health questionnaire that we distributed at the beginning of the study. And as I previously stated, it just contained items such as age, gender, race, and then asking just for a general health background, so asking if the participant had any of those conditions such as gout, diabetes, diuretic use, or none of those conditions, and then also inquiring whether or not the participants had a family history of gout, and then also how often they consumed sugar-sweetened beverages. So in this study, we enrolled 13 participants total. However, the data was analyzed per protocol, so it ended up just including 5 males, 5 females with ages ranging from 18 to 37. Three participants were excluded from analysis due to either incomplete dosing or vomiting. And the mean salivary urate levels was found to be 3.74 milligrams per deciliter. That was 1 hour post-ingestion. And so you can see that that is still falling within the normal range for uric acid level. Urate levels peaked at 2 hours following fructose load for both heterozygous and homozygous groups, as indicated there, with the heterogeneous group being just slightly higher than the homogeneous group. And urate levels did not return to baseline by the final collection time. So this table just shows the breakdown of our participants with homozygous versus heterozygous genotypes. 3 out of the 10 participants did show the variant allele, meaning that they just show the presence of this polymorphism. And this is just a figure showing each individual urate profile following the fructose ingestion. So as you can see, it's kind of all over the place. This includes also the participants that did show the variant. It's indicated in different colors. It might be a little difficult to see. However, I wanted to point out that the green line here, if you can follow along with the laser here, and the brown line, as well as this gray line here. Those individual profiles are the participants that did show the presence of the variant. Just to reiterate my point earlier that it is kind of all over the place. And I wanted to just quickly highlight that as compared to the homozygous group. Heterozygous was slightly higher, but there was no statistical significance as indicated by the p-values on the right-hand side. So when we looked at the urate levels for each group at the respective time points, half hour, 1 hour, 2 hours, and 3 hours and 4 hours, there was no statistically significant difference between the urate levels. This is a graphical representation of the same information that I had just discussed, with the lighter blue line representing the heterozygous genotype up top and the homozygous genotype at the bottom. And as you can see here, the urate levels peaked at 2 hours following the fructose ingestion, and it's indicated by the standard error bars as well as the p-values previously discussed. There was no statistically significant difference between the two groups in urate levels. This figure here is a time average concentration of urate comparing the heterozygous and homozygous groups. And as you can see here as well, there was no significant difference between the two groups as far as systemic exposure to urate levels. So overall, we found that urate levels peaked after 2 hours following the ingestion load for both groups. There were no statistically significant differences in urate levels between both groups, as indicated by the p-values previously discussed. And in conclusion, our results do not support our hypothesis that genetic variation in the SLC2A9 gene impacts systemic exposure to urate following fructose load. I just wanted to discuss a few limitations to this study. Of course, it was a small sample size with only 10 participants enrolled, considering that it was a preliminary study. Also, we had a short duration of study, so the total duration of the collection was within a 4-hour time span within 1 day. And we also did not have a negative control. As far as opportunities for future study, enrolling a larger sample size would help to provide further data for analysis. Also, considering an extended sample collection time to see how long it would take for the participants' urate levels to return to baseline, because that was not something that we observed due to the fact that we stopped collection at 4 hours. But as you may recall from the graphs, there was a trend going downwards after it peaked at the 2-hour mark. And lastly, considering an additional medium of collection may be beneficial just to have something else to compare to. So for example, perhaps also collecting blood samples to see if salivary urate levels are comparable to serum levels. And lastly, I would just like to thank my research advisor, Dr. Maturka from the Gregory School of Pharmacy, for his mentorship in this study, and also the Gregory School of Pharmacy, specifically the Department of Pharmaceutical Sciences, just for the lab space and opportunity to participate in the summer research program, which was a great experience and just a great time. These are my references. And I thank you for your kind attention and for being here, and I'd be glad to answer any questions that you might have.
You analyzed it like each half-hour interval and compared the groups that way, or?
Correct.
Okay,
So we
you
just,
did t-test like at half-hour, 1 hour?
correct.
Okay. Did you consider maybe doing like a mix? You probably wouldn't have found anything given the variability, but doing like a mixed ANOVA, something along those lines, where you had like groups as one factor and then time as the other factor?
Yeah, that wasn't something that we were focusing on in this study, so we just strictly compared the two different groups depending on whether they were homozygous or heterozygous.
Yeah, I wouldn't give up on it. I think with a larger sample size, you might find something.
Was there anything about, you did a super job on this project. I really want to congratulate you for that. part of the experience that was particularly notable to you?
Of this summer research program?
Anything you learned or anything you're taking away from it? You know,
I would say that the biggest learning opportunity from this experience was just learning how to design a study, because we had actually written a protocol, very elementary, but a protocol that we provided with each of the participants so they knew exactly what they should do to prepare for the collection. And also the entire research process of designing a study and recruiting and administering the samples and collecting the samples. It was just overall a good learning experience.
I think you put together the IRB form too, or you were involved in that?
Yeah,
You did get IRB approval for that.
any other questions? All right, thank you.